Click for IB Biology. We're in the genetics topics and we're looking at the concept of dominance and recessiveness in the interaction of alleles for a single gene. There's a blank version of this presentation, a PDF, linked down in the description below. You can download that and annotate that with your own notes as we go along. But then at the end there's a quiz and there's 10 questions. You do those and check out the answers to make sure you've understood the content of this video. Okay, so we have a chromosome, and the example of the allele that I want to talk about is for the gene for a taste receptor. And this taste receptor, which is on our tongue, allows you to taste a chemical called phenylthiocarbamide, or abbreviated more conveniently to PTC. Now, there essentially are two versions, the two alleles for this, which is gene TAS2R38, apparently on chromosome 7. There's two alleles, and one which allows you to perceive a bitter taste, and then the other allele gives no taste. So let's look at the occurrence, the possible combinations of these two alleles in individuals, remembering that you will have two versions of every allele. So the first one I'd like to discuss is that we inspect the two chromosome 7s, and what we find is that the version of the gene there is the big T one, which means that we have a bitter taste receptor on our tongue, and then we look at the other one inherited from the other parent, and it turns out that they are identical and they allow us to taste the bitter polythiocarbamide. Right, so the genotype using the letter system is expressed as big T, big T. And the phenotype would mean that the individual here would be a taster. So they can taste the bitterness. So that's this individual here with their homologous pair. The alternative, and if you don't mind, I'm going to do it down at the base of the page. The alternative obvious combination is that if we look at the same gene loci, we find the little t version of the gene, and here there's the little t version of the gene, and so the genotype of this person is little t, and that person would actually be a non-taster. Notice that for the one that we've done here and here, these are both homozygous, that their alleles are identical. Now the interesting event occurs where in the third possible genotype, where we have the presence of big T on one of the genes, one of the chromosomes rather, and the other one is the little t version. So in this particular case, and they have to be studied on a case-by-case basis, the genotype then would be given big T, little t, which of course this is the heterozygote. And in this particular case, what happens is that the gene version, big T, when expressed, completely masks the effect of the little t. So it's completely covering up or masking the effect of that little t allele, and therefore this person is a taster. So let's make a couple of points about this. The allele which is covered up by another allele, we call that one recessive. And the allele which does the masking and the covering up, we call that the dominant allele. In this case, there is no sign of non-tasting in the heterozygote. So this is complete dominance that we have here. So the heterozygote is a taster individual. And so if we look at the summary of what we had here, we had one gene, the gene for tasting. So that's what we call monohybrid condition. We had an example of dominance where big T dominates little t. We had 3 genotypes, but we only had 2 phenotypes. And so within the population, phenotypically, the group of people in the population who are tasters could be homozygous dominant, or they could be heterozygous. Those who are non-tasters are definitely homozygous recessive, little t, little t. So this is an example of monohybrid genetics with complete dominance. The link to the next video is to the phenomena known as codominance. The channel link is there. You can subscribe. That would be greatly appreciated. Some likes would really be appreciated as well. And the playlist for all the other genetics videos. Thank you for watching.